pleasure to introduce today's second presentation, What's New in Gene Therapy Across Neuromuscular Diseases. And we are so honored to have Dr. Byrne here with us today. Dr. Byrne is a clinician scientist who is studying a variety of rare diseases with the specific goal of developing therapies for inherited muscle disease. As a pediatric cardiologist, his focus is on conditions that lead to skeletal muscle weakness and abnormalities in heart and respiratory function. His group has made significant contributions to the understanding and treatment of Pompe disease, a type of neuromuscular disease due to glycogen storage in motor units. The research team has been developing new therapies using AAV-mediated gene therapy to restore cardiac and skeletal muscle function in Duchenne, Friedreich's ataxia, Pompe, and other inherited neuromuscular diseases. His group at the Powell Center has also established a series of new methods for large-scale AAV clinical manufacturing, and this work is supported by NIH, industry, and foundation awards. Dr. Byrne is also the Associate Chair of Pediatrics and Director of the University of Florida Powell Center and Child Health Research Institute. He obtained his bachelor's degree from Denison, his MD and PhD from the University of Illinois, and completed his pediatric residency and cardiology fellowship, as well as a postdoctoral training in biological chemistry at Johns Hopkins Hospital. And following his early career at Hopkins, he joined the University of Florida and now is the Earl and Christy Powell University Chair in Genetics. He also serves as the Chief Medical Advisor here at the Muscular Dystrophy Association and is a member of the TAC Committee of Treat NMD. So thank you for sharing your time and expertise with us today, Dr. Byrne. I know we went a little over, so don't rush. Feel free to take your time. We definitely want to hear from you. So I'll turn the time over to you now.
Thanks, Marissa, and thanks so much for the kind introduction, and thank you all for being here. And so it was hard to follow Paul and the great work that's being done in the advocacy organization at MDA. So let me go ahead and share these slides and start to go through some of the things that I think will be of interest to this group, and appreciate you all sending questions at the time of registration. And one of the topics that kept coming up is, how can we make this process faster? So I did ask Paul to share with me one of the topics he's been working on. I'll touch on that at the end. And hopefully what I'm covering is generally relevant to each of the gene therapies that you all ask questions about, whether it's DM1 or limb-girdle muscular dystrophy, FSHD, the various specific indications where there's been a lot of work in gene therapy. Obviously, we have paid close attention to the work in Duchenne and now the recent authorization of ELEVIDYS. So here's a bit of disclosures. Marissa mentioned I'm really an honor to be part of the MDA as a medical advisor and recent board member. And I'm not going to talk about any specific products during this presentation. So here's the general outline of what I'd like to cover. To give you a little additional background, because often we just jump into this as though it's a thing. And it's important to realize how far this field has come in a very short time. This was, when I started my medical training, this wasn't a topic. And even after I finished fellowship, this wasn't a topic. It really required some of the early stage laboratory work that was first done at our institution now at the University of Florida that developed the foundation for this. And I just hope to give you some encouragement that the rate of discovery in this area is increasing at almost an exponential level. And it's I think, important to support the current therapies as well as the next generation of therapies. I want to talk a little bit about eligibility for research versus commercial use of a gene therapy product, what it means to have a gene therapy product that is essentially going to permanently change some aspects of your genetic makeup, hopefully in a favorable way, but that influences ability to participate in clinical trials and for other products, and I'll talk a little bit about the notion that you could receive another gene therapy product later as well as some of the things that we've identified as safety concerns in the way gene therapy acts. So this is where I work at the University of Florida, Northern Florida. The region of Northern Florida, population of about 12 million people, many of whom travel here to receive health care. And as it relates to gene therapy, we actually see patients from as far as Atlanta, which is all the way to the right side of the screen, all the way down to Miami, which is on the left side of the screen. And one thing we do have, even though we don't have mountains in Florida, we do have lovely sunsets. So you see out west is the Gulf of Mexico, you can see from our area. We're really in the middle of the state. Intentionally, the university was placed there to be accessible to all the major cities, although it does sometimes make it a pain to get there as a study participant because there are no direct flights other than from Atlanta or Charlotte. But our goal in the health system is to really innovate in care for rare and genetically defined diseases, which in fact covers most neuromuscular diseases. So internally, we rely on the Children's Hospital as well as our new cardiac and neuro hospital for partnerships and the growing regional network that exists in Florida, particularly also for newborn screening, where one of the key sites that identifies patients with genetically defined diseases like SMA, for example, where early intervention is very important. So that means we engage the community around us and in the state to learn about and participate in what we're doing. I mentioned the newborn screening program, and I'll show you a little bit later the complexity of working across various academic institutions and industry partnerships to develop products that will help improve the lives of people with neuromuscular diseases. So we, of course, have to have some clinical expertise. The university's made a huge investment in artificial intelligence and machine learning. So those analytical methods are going to be very important for understanding disease variability within a given class of diseases, as well as adverse events and responses to things like gene therapy. And interestingly, we're using machine learning to actually design better AAV vectors that have improved ability to reach the key target tissues that are relevant in neuromuscular diseases like the brain, the heart, the musculature. And that's where translational research comes into play. We, of course, as part of that, have to train the next generation of clinician scientists and basic scientists who we really begin then all over again when we develop additional people with this clinical expertise. Because this field, actually, I'll make an analogy later to transplantation, didn't exist really for many people who are in training now. It's kind of a fantasy. They think, well, there's maybe be gene therapy in the future. We say, no, it's really happening today. And luckily, most of the time, these events, encounters with patients, are all done in the outpatient setting. So I'll show you a little bit how that happens. So our trainees don't always get exposed to this technology and how it can be transformative in the life of someone with neuromuscular disease. So DNA, what does DNA do, and how did we ever dream of manipulating DNA? And really, the concept of how inheritance was transmitted through DNA and how DNA carries the instructions for making up all the components of the cells in the body that are the complexity of a living cell and a living organism. It's present in every thing from bacteria to humans, and it provides all the instructions for what cells do day to day to provide the function that they do within the complex organism of a person. And now we know that there's not only ways to add DNA back, but to change it, as in this slide. In some cases, a single base change may confer a disadvantage to a given gene or an abnormality in function. So this was the mission, is like, how do we manipulate DNA? And potentially the simplest strategy would be to add back genes. So genes particularly unique about dystrophin is they can be of various sizes. Some genes are very small and some genes are enormous. In fact, dystrophin has the largest gene in the body, but for the second largest protein in the body. This is made up of a number of exons that code for the protein, and the protein is made up of amino acids. So proteins that we eat get turned into, from the amino acids they're made of, get turned into proteins that the cell needs to function. And the way we learned now about 30 years ago by investigators at UF, DNA can be loaded into this small viral particle called adeno-associated virus. And in the beginning, honestly, we really didn't know how this simple virus actually lived, what its life cycle was like. And so much of the early work when I first came to UF was to understand how the natural life cycle works of the virus so that we could hijack that process to be able to make it as a drug. And so we know that even though AAV itself doesn't cause disease because it's part of a group of viruses that depend on other viruses to work, but it can provoke its own immune response. And we've seen that in a number of cases where it's used therapeutically. One of the things that came out of early work done at University of Pennsylvania was in the early 2000s, we learned that there were not just one kind of AAV, there were hundreds of kinds that existed in nature, and each one of them had a specific different capability. And now we, using this machine learning artificial intelligence strategy, can take advantage of these unique features. So these parts of the virus that are protruding here are where it attaches to cells to get inside. And because of really brilliant work done by one of our faculty in biochemistry, Mavis Bench McKenna, discovered this structure using a special imaging technique and determined that it's possible to put structures here that improve the ability of the virus to get inside cells. So I'm going to talk a little bit about that because that's really one of the great opportunities for future use of AAV vectors. And certainly so far, with the few products that have been approved, in considering risk versus benefit, this really favors the therapeutic use. And I mentioned Mavis and the other AAV pioneers like Nick Mazuska, but that's recent analysis of the worldwide literature of AAV. So you see, we seems like it represents a lot of work, but there are 5,000 articles that were analyzed since the beginning of the first publication about AAV, and you can see how this rate of publication is increasing very quickly. And thankfully, this red cloud shown here shows that our university has contributed more than any other to the worldwide literature about AAV, and we hope to continue to train new people that go out into this field and continue to publish on this area and improve the field and make, of course, these more accessible and safer. Just a brief history, you know, even the idea was not really born until 1990, but it was really in the late 90s that the first AV work was being done, partly in our group and partly at the NIH and at Nationwide Children's Hospital, where a small group focusing on spinal muscular atrophy developed the first approval for spinal muscular atrophy. And this, along with the approval for the inherited blindness drug Luxturna that was developed at UF, this stimulated the biotech and pharma community, who had almost zero interest in AAV prior to that, to get involved in helping to develop AAV products. And so now, keep changing the wrong way, sorry about that. Now we have approvals for hemophilia and Duchenne, as I mentioned. And what comes next? What does the future hold? What do we need to do to make this better and more accessible and safer? One thing for sure that we're very focused on is how to make the products at a lower cost. This will lower barriers to access, not just in the US, but outside the US, where it's more difficult to get reimbursement for high cost products. And right now, even though these costs seem astronomically high, they're actually that high because it costs a lot to manufacture the product in a safe and reproducible way. So improving the innovation in manufacturing hopefully will lower costs and improve access, particularly for the smaller population. So the commercial viability of a product is influenced by the population prevalence. And so this is why Duchenne was one of the greatest interest, because there are up to 10,000 patients in the US with Duchenne, and we see development happening very aggressively across a variety of sponsors, especially with one that's reached marketing authorization. I put in here a few years from now the possibility of multi-vector design, and this will expand the utility of AAV for things like base editing or CRISPR-based gene editing, and also the potential to make larger dystrophins, which would be potentially more effective in Duchenne. This is really ultimately the goal, to continue to innovate ways to improve on the products that exist today to make them better tomorrow. And so I mentioned editing. There is really the holy grail, particularly in many neuromuscular diseases that affect predominantly the muscle. Since muscle cells continue to grow as we get bigger and older, and cells are replaced by new cells, those stem cells that provide the ability to muscle to repair itself. And if we could correct the gene defect within the stem cell while it's in the body, not like in bone marrow transplant, the stem cells are removed outside of the body and done and used in what's called ex vivo gene therapy. If we could target those muscle stem cells, we would be able to provide correction that would really last lifelong. So that's the goal. And then it's important to say, for the advocacy that's being done by MDA, the regulatory framework is one of the aspects that contributes to cost. And so much is being done to help harmonize regulations across the US and the rest of the world to, again, improve access and lower cost. So let's just talk about the current status. So here we've come a long way. That first gene therapy approval with an AAV vector was Luxturna in 2017, developed at UF and University of Pennsylvania. And then the important work done at Nationwide for Zolgensma was the first systemically delivered gene therapy, followed by the approvals in 2021 and 2022 that ultimately led to hemophilia gene therapy being authorized. And now, at least in 23, the first approval for Duchenne for the young children, as well as 2 exciting gene therapies for sickle cell anemia, which is a much larger population. So this is, as you can see, this list is getting longer, and that actually helps us learn more about other conditions. And we learn a lot from one program to another. This was the first baby to receive commercial gene therapy at our institution, and it was just a matter of a unique timing of diagnosis and availability in 2019. Now all babies being treated are really within the first month of life. And in the US, there's been about 1,000 children who've received Zolgensma and about 2,000 in Europe. So we learn a lot from that product initially, and it's teaching us how to apply those same principles in Duchenne. And one of the things that I think can't be denied, and the FDA talks about this a lot, is that whereas an infant with SMA might never have sat independently and certainly not stand alone, they would not walk and often be dependent on a ventilator for their life. And so we know that treatment with Zolgensma is transformative. There are aspects that we still need to learn about that may impact sleep, swallowing ability, are some things we're still learning about, but there are additional treatments that are added to gene therapy later if needed. So we have a toolkit that at least arrests the problem as soon as it's identified in a newborn. And then we continue to follow carefully these patients throughout their life. And, how do we do that now for Duchenne and all the other muscle diseases that may not be permanent because muscle cells grow, unlike neurons are present when we're born and they don't change over the lifespan. So this was an important milestone to get Elevidys approved, and really just now embarking on the journey of going beyond this 4- and 5-year-old age group, keeping in mind that in the beginning of the approval, half of the 4-year-olds were not even diagnosed yet. So now we're catching up on those when they become clinically evident, and the average age of diagnosis is still about 4.5. And of course, this is a great opportunity for the non-ambulant population where outcome measures were more difficult, and there had been very few products that were available to that age group. And we've divided our group into 2 groups that really represent one team, those that are doing the preclinical discovery work, and the clinical teams that both do research and clinical care. We really work seamlessly together, and oftentimes the way we learn is by taking things that we observe in the clinic back to the laboratory again and trying to improve on it. Just show you that there's a large group that does this, both the preclinical laboratory scientists as well as the partnerships we develop with other organizations, either federal organizations or funding entities like are shown on the left, or industry. And you can see the number of industry relationships we developed to be able to provide the clinical studies to the patient population, as well as developing academic collaborations shown here on the right. So this is a complex set of activities that are basically constantly changing. And so we use that now to take those things to the clinical research team. And so here are the people that actually run the studies, and that often become family members with those who participate in the studies. We even have a comfort specialist who's Bailey's, Mackenzie's dog, who's a therapy dog. Of course, we need a place to do this, and the care center network is where this happens predominantly. I encourage you, if you're on this webinar, you know about MDA, but you can spread the word to people that you think may need support and information to go to MDA and get that information. A lot of the questions that were put in at the beginning were about how do I participate in a clinical trial and how do I find out about them? Well, MDA tries to curate some of that information, but it is required of a sponsor to also report this to the at the federal level through clinicaltrials.gov. This is a little clunky tool because you can't set up a search that comes back to you automatically all the time. I think they're trying to work on that, but you would go to this site and put in a specific indication, and this site will list for you all the active studies for that problem. And it will help you add terms to the search, like if it's just Becker muscular dystrophy, you can use that term, or muscular dystrophy in general, you're going to get a lot of different studies. So the site can narrow down certain conditions and locations, and sometimes it's actually smart enough to even know where you're searching from and point you towards through the Google search engine sites that are in your area. So that's how you find a study. Then how do you participate, and what are some of the criteria, particularly as it relates to AAV gene therapy? So the first gate, even for newborns with SMA, that they have to go through is to know that there's been no environmental exposure to the gene therapy agent, which is a virus that exists in various forms in nature. I will say something about this related to SMA because it's a very important issue that's unfortunately got some conventional wisdom that wasn't exactly accurate. When a baby tests positive for AV antibodies, it was in some cases assumed that was related to the mother's breastfeeding. And we have a paper under review right now that shows that really it was related to the placental transfer of the antibodies, and breastfeeding does not transfer antibodies against AAV, and that one should then, there's an approach to use to wait for those antibodies to dissipate until that child were to become eligible for gene therapy, but it's not through breastfeeding. So when that paper comes out, we'll distribute it, and it'll have further evidence of the way to approach positive antibodies in newborns. Those generally go down by about a year of age for sure, and mostly diminished by even 6 months. But one of the things I wanted to emphasize is that when you become eligible for gene therapy, once the gene therapy is delivered, and particularly if muscle's the target, this is a lot like organ transplantation, and we're really learning some of the immunology around gene therapy is a lot like we learned many years ago in organ transplant, and that's actually how I started my career in heart transplant. And so this is one of the two medical therapies that exist that are in effect permanent. So that's something to keep in mind. And what happens when we give gene therapy? So the infusion here is on the left. There are a lot of mechanisms we have standing by in our body to reject infections like from viruses. So they can be pre-existing antibodies, like I just mentioned, or antibodies you develop in the days following the exposure, and then white blood cells. And another function in our blood is called complement. And we know now that the complement system is probably a double-edged sword for AAV. Parts of complement are needed for the AAV to work. And when it's out of control, though, it can create a safety problem, and I'll touch on that. And then ultimately, those are the at the ready immune response called innate immunity, but there's also acquired immunity that takes weeks or months to develop. And it's really because of this acquired immunity that a very high level of antibodies are formed that prevent another use of AAV. And just like we would be immunized against a certain early infectious pathogen to prevent illness, the AAV exposure prevents a reinfection with AAV. And I'll refer you to this paper, which is a plain language summary of more in-depth technical details about the antibody test itself, because we get a lot of questions in the clinic about this, about when are antibodies developed, what can be done about it if you're not eligible for a gene therapy. And in fact, we're working very hard on a study supported in part by MDA and other organizations to address the issue of pre-immunity in patients that don't qualify. But this plain language summary has a number of great illustrations and is a lay summary of how the antibodies are tested and what it means. So testing for antibodies is by two different methods, and each sponsor decides what method is appropriate. The binding assay, which is shown here in the first part, is what's used for Zolgensma and Elevidys eligibility. The Pfizer product that was being developed for Duchenne used the neutralizing antibody assay. And they both essentially assess whether there's any detectable antibodies that bind to the AAV, the specific AAV that's being used. In general, we do find that when there's some immune response to one AAV, they're also closely related, that there's the same immune response to other AAVs. So it wouldn't work out that if you just have an antibody to AAV9, you're likely to not have antibodies to anything else. One good thing about the way Elevidys was developed is that the threshold for allowing to receive the product is higher than any other AV product so far. So that means more people are included. And in our real world experiences, about a third of individuals have had some pre-existing antibodies, but they're very close to that threshold. So we usually also repeat the test and consider what other studies might be available to that person while working out how to lower antibodies. Here's what we found over the past 5 years of patients that were seropositive at baseline, not for any one product, but just in general. We've tracked them now every 6 months for 5 years. And you can see that once you're seropositive, the cells that make the antibodies continue to make low levels of the antibodies. And this threshold of about 500 units is what's excluded from studies. If you get closer to zero, those are the ones that are all included in studies or commercial products. So we see the antibody response, once it exists, is quite persistent. In fact, this yesterday was the 25th anniversary of the first patient who received an intramuscular injection of AAV. Donovan Decker has limb-girdle muscular dystrophy, and he posted about this yesterday. It was the exact day 25 years ago, and he's had antibodies tested, is still positive. So we want to figure out ways in which early volunteers of studies like him could access products as they are improved over time. So here's what we're considering as ways to treat seropositive patients. It's possible to eliminate the cells that make antibodies. That's the first strategy that we've used in several studies now. There are strategies to change the way the AAV is built to make it go more quickly into muscle cells and avoid antibodies. We can try to eliminate the plasma cells. That's the third strategy. And now there are ways in which we're using decoys to reduce the affinity of the antibody for the AAV or deplete the antibodies from the circulation temporarily. So these have been tested in patients. We hope to run this larger study repurposing a drug used in myasthenia gravis to treat those that are very close to the threshold. Those drugs lower antibody levels about 80%. And we think that might be sufficient for the majority of cases. Keep in mind that AAV will be used for other things in the future. We mentioned gene editing. So that's shown up at the top here as CRISPR. The way it's being used now is as it could deliver a transgene like microdystrophin. We can imagine whether multiple AAVs can be used to actually deliver full-length dystrophin, or even ways to block a dominant gene or do skipping via a vector administered that would make also longer dystrophin. So there are lots of future ways AAV will be used, and we'd like to make sure that as these new modalities become available, that people are eligible to receive them. I want to say the MDA is doing a lot in that role for advocacy. Last year, I just show Nora here represented the MDA at the Advanced Therapies Week. MDA won the Patient Advocacy Award on behalf of the biotech industry. And as I mentioned, Paul provided me with this slide about the Priority Review Voucher Program. I don't know whether he had the chance to cover this in the last session, but this is very important to see reauthorized because it's a stimulus for many biotech companies to develop products, because they can use this ticket, if you will, to get a discount on future program development. And so we hope this continues to be authorized through Congress, acting very soon to reauthorize it. I'm going to play this video about one of the goals we've had about, I mentioned several times, redosing. So we have worked with the KOMD Foundation to develop a strategy for redosing AAV, and this is just a little video they prepared about that and why it's important to them. And Connor, who was one of the first participants in the Pfizer study for Duchenne. So this is obviously really important to many families, particularly those that were the earliest participants in studies. So I just hope that this brief time I've been able to cover the fact that, AAV gene therapy for many neuromuscular diseases is going to be really transformative. I hope we can get the level of efficacy seen in SMA in every neuromuscular disease, and maybe we'll even call those curative treatments. There's a lot to do to make that happen. The transformative nature of it relies on collaboration across industry, across academic institutions, and the teamwork that we have and partnering with MDA. And of course, this big challenge we have before us relates to manufacturing, managing immune responses so that so that gene therapy is safe, first and foremost, and that we can use these like conventional drugs and maybe find not everyone will receive the same dose, and we have a mechanism for incremental dosing that we're imagining. So that's what's a priority for us right now, and we've tried to bring all this together in a new center called Advanced Therapeutics. This is where all the commercial gene therapy will be administered on the first floor. This is immediately adjacent to our research building. And so we're just now opening this center as the commercial therapies become available for Duchenne and for SMA. And I'll end there and take questions that anyone has. We finished on time, maybe.
Yes, thank you so much, Dr. Byrne. I'm just incredible amount of information and so many exciting advancements. I've been with MDA for 11 years, and just that change that I've seen, the acceleration of all these things is amazing. So thank you. We do have some questions that came in, so let me just get to those here. We had somebody ask where they could access longer term results from when gene therapy started to now. They gave the example of trials for Elevidys possibly starting around 2017, 2018. And being able to see where those kids are now.
That's a great question. Sarepta does try. So once a product's authorized, most companies don't end their efforts there. They continue to follow patients enrolled in observational studies long term, and there is a long-term observational study being done of both the study participants and even potentially some of the individuals who received the commercial therapy. And so they will continue to present those at medical congresses, and Sarepta does a really good job of making all those presentations publicly available. Maybe we can distribute at some point the site they use. It escapes me the exact designation of the site, but it used to be hard to find that information. They've been very good about making it publicly available, as our other sponsors now, because they need to continue to educate the provider community and the patient community. So there is supposed to be also updates on clinicaltrials.gov. Not every sponsor does that in a timely fashion. So it really depends on which product you're trying to learn about. But and then there's no reason not to reach out to a sponsor that's developing a product and ask them directly for what's the latest in this area. They often have non-confidential material they can share, or they would point you to an investor presentation. I mean, part of the reason they disseminate this information widely is they have to maintain interest in the public to the products they're developing. And so they are required to do quarterly updates, and most of the time those quarterly updates have a lot of information that the public can access.
It's great to know that. I wouldn't have thought to reach out to them directly, so that's great.
It's usually through their investor page, so that's a place that patients shouldn't be afraid to go look for updates that come from the company.
Great, good to know. We are getting questions in the chat about specific diseases. It's always hard to cover all of them, but you mentioned FSHD. Can you speak to any progress in that area at all?
Yeah, so it is a little more complicated when there's a dominant condition, and there is a lot of work being done in this area, both through gene editing and gene silencing approaches, and but also using AAV. I shouldn't say that everything is based on AAV as well, because there are clever strategies now that can put therapeutic proteins into cells, particularly like muscle cells or liver cells, and they will secrete a therapeutic protein into the bloodstream, or even using the same way we all had vaccinations against COVID, using RNP mRNA. So mRNA therapeutics are still very active, and you can imagine ways to add or reduce a gene function through those methods as well. These use lipid nanoparticles as a way to deliver the active product, and those could be used to reduce a gene function.
What about, we had a question for OPMD and another one for LGMD. Anything specific you can share around
I'm not aware of any gene therapy studies for OPMD. So no, not in that area, but really any gain of function, loss of function is amenable to gene therapy. And it really is just the imagination of the investigator that wants to pursue that problem. And it's often stimulated. I can say in my experience, many of the programs that we've begun were first brought to our attention by a parent who said, why not this problem? And we'd say, well, you're right, let's get to work on that. And it's really, that is an important motivator for families to bring those ideas forward and help.
How would they do that? Would they go to their clinician? Would they go try to find a researcher?
So this is where you kind of have to partner with your primary provider at your care center and say, well, I know we're taking care of the usual clinical problems with this condition, whatever it is, but ask who is the person that is leading research in this area? Can you put me in touch with them? Because, among the care center network, everyone has a different level of contribution in different areas. Some specialize in even just one form of limb-girdle, or another person works on just cardiac issues. But together, the collective wisdom is really amazing. And so it's just a matter of asking at your care center, how do I get in touch with so-and-so? And it sometimes requires slogging through the literature a little bit and see who's generating the publications around a given topic. And then I would say once a week, I get an email from a parent who has a question about a unique situation they're facing with their child, or for someone like Donovan who's had gene therapy already.
Another question came in. How likely is it that there will be a better gene therapy than Elevidys for Duchenne?
Well, we do know now that there is the ability with multiple AAVs to make a full-length dystrophin. We also know that there are strategies to get, even though the amount of the percent of cells expressing microdystrophin is very impressive, in almost half of the muscle cells are being protected to some degree by Elevidys. But we'd really like to get that to all the muscle cells, all of the heart muscle cells. About a third of boys have some inattention and learning problems because of the defective dystrophin in the brain. So there are areas where we can improve. So I'd say that it's very likely that there'll be improvements. It's the unknown part is when. It will be harder to manufacture those longer dystrophins. There may be additional immunological findings with multiple vectors that really in effect somewhat of a higher dose, or ways to manufacture those multiple vectors. So, but as we learn from the programs that exist today, that'll help build a stronger foundation for the programs that will be started in the future. And so I think we have to keep striving to improve each of these therapies until they're curative.
Definitely. Some of the questions that came in through registration, I noticed there was a theme a little bit there of is gene therapy only for the younger generation? Do you see in the future it being possible for older individuals to benefit?
I do. I think we can't forget about those boys and young men who've lived with Duchenne for so many years. And the big question that many providers face in that is how to be sure it's safe. So, as the dose is based on weight, the larger dose is associated with more safety events. And so we have to go cautiously in those older individuals because it really was a very small group that have received the product thus far in the authorization of ELEVIDYS. So as more experience is gathered, and it's not just from Duchenne, I might add, in one area where we're learning a lot is there are several sponsors working in gene therapy studies for inherited heart disease. These are generally adults. Sometimes they're dominant conditions where they have affected children as well, but in general, the studies are being started in adults. So they're in some ways the worst case scenario for these adverse events. And we're learning how to maximize the expression in the heart because they can consent to heart biopsies that are telling us about the degree of effectiveness of the current products. AV9 and RH74 are being used in these programs. So we're learning from those studies what we should be aware of for adults with Duchenne or Becker, or even manifesting carriers you might imagine someday would be amenable to these treatments. And so safety first. And once we have that figured out, I think we're really going to try to prioritize access for those who have waited so long to get some meaningful therapy.
Well, I think that's a kind of a great, hopeful place to leave it. And thank you so much, Dr. Byrne, for your time and preparation and expertise. I think this is such wonderful information. So we appreciate you being here today.
Glad to join. Thanks, Marissa.
Thank you. So just over a year ago, as Dr. Byrne mentioned, we established our gene therapy support network. So I just wanted to share a little information about that. It does include a phone number and email that you can reach out to and schedule a one-on-one video call with one of our gene therapy support specialists. We also have resources on the website, a whole learning module that takes you through gene therapy and how it works. We also have educational materials both in English and in Spanish that you can print out or send digitally. And we also offer virtual learning like the webinar that you're on here today. Also wanted to just let you know about our community groups. This is another relatively new program, but there is a supportive group specifically for the gene therapy community for parents and guardians of children who have received therapy either with Duchenne or SMA. We also offer groups outside of gene therapy that are just for parents or for adults living with neuromuscular disease and for our ALS community. So would love for you to join. It's a great place to get support and to just share resources with others. I wanted to just remind you to join us next Thursday, September 12th, for the last part of our gene therapy series. We'll be talking about what questions to ask your provider as it relates to administering gene therapy to make sure where you're going is well equipped. And then we'll end with a panel discussion with families who have gone through this process and learn from them about their experiences and what factors they considered when they were deciding. So please scan the QR code or click on the link in the chat and you can register for next week. And we'll also be hearing from Dr. Panda from Arkansas Children's Hospital. And then Lastly, wanted to just invite everybody to our in-person symposiums that are happening this year. If you live near Georgia, California, Texas, we are meeting in person and having some wonderful clinicians join us to provide education, not just on gene therapy, but on many other topics related to neuromuscular disease. We do offer travel stipends if the cost of traveling is a barrier, but we would love to see as many people there as possible. And then lastly, if you're not already a part of MDA, please join our community. We'll put a link in the chat, but this is free. It's free to join MDA, and it will help you stay connected to other individuals in the community, as well as the latest in clinical trials and just helpful resources for the community. We again would like to thank Dr. Michael Story, Paul Melmeyer, and Dr. Barry Byrne for their time today. We want to hear from you. What did you like about today's program? What can we do differently? Please fill out the survey that will pop up right after the webinar. It's very important to us. Your feedback is really helpful. And if you have any other questions, you're looking for support, our resource center is a great place to go. The phone number is there on the screen. We'll also put it in the chat, but please reach out to us anytime and we will be happy to help you. And with that, we'll say that it concludes today's webinar and wish you all a great night. Thank you again for attending.